Hello everyone, and welcome to today's live broadcast, Perspectives for Whole Cell Microbial Reference Materials, presented by J. Russ Carmical, PhD. We are excited to bring you this educational web seminar presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. I'm Julie Simmeroff of LabRoots, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window or use the Q&A button to let us know that you're having a problem. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. Now I'd like to introduce Dr. Carmical. Russ is the Sequencing Director of the Alkett Center for Metagenomics and Microbiome Research at Baylor College of Medicine and Director of Operations for Diversigen, where he applies laboratory automation to metagenomic and microbiome sample processing for next-generation DNA sequencing. The laboratory is CLIA CAP accredited and offers regulated services to both academic and commercial collaborators. He is also one of the founding members of the International Metagenomics and Microbiome Standards Alliance, which was formed for the mutual benefit of the entire microbiome and metagenomics community and will focus specifically on coordinating cross-cutting efforts that address microbiome and metagenomic measurement challenges. Please join me in welcoming Dr. Russ Carmical. I will now turn the presentation over to him.
Thanks, Julie. As Julie said, I'm Russ Carmical. I'm the Director at the Center for Metagenomics and Microbiome Research at Baylor College of Medicine. We're located in the center of the Texas Medical Center. I'm going to talk to you about whole cell microbial reference materials in regards to metagenomic sequencing. So, because we have such a diverse audience, I'd like to define a few terms before we dive into that. And the first would be the microbiome, which is the entire collection of bacteria, viruses, and fungi, and naturally occupying various niches on or in a given environment. And the second would be metagenomics, which is the use of modern technology, in our case, next-generation sequencing, to study communities of microbes directly in their natural environment without the need for isolation or cultivation. And this is where next generation really advanced to this field of study. So this kind of set the context for the driving forces behind what we're doing. In addition to academic collaborations, which are handled through our service center, the CNMR, we have a commercial arm which services our commercial partners, and that's Diversigen, which was founded in 2014. And so the mission and the vision for Diversigen is to apply metagenomics to the benefit of human and animal health worldwide. So as Julie said, we're fully accredited, both CLIA and CAP accreditation, and I'll discuss a little bit about the accreditation process and why we embarked on that. So I think this statement here really underscores the need for accreditation. That is that if metagenomic analysis are to evolve into a mature clinical diagnostic, then an appropriate accreditation program ensures quality testing, but most importantly, patient safety must be developed. And so that's what we set out to do, to not only help our laboratory, but the field as a whole. If you do a literature search, you'll see an ever-increasing number of researchers that are conducting metagenomic studies with a clinical endpoint. When I first gave this talk, I struggled to get a couple articles on the slide, and now we can fill up many, many slides with the number of studies that are being conducted with these endpoints. So in addition, if you were to do a search for companies that are conducting metagenomic studies, they're ever increasing as well. And then, of course, we have the buy-in from the national government with the announcement of the National Microbiome Initiative back in 2016. So again, just kind of set of the context for the talk. I'm going to speak specifically about 16S sequencing and profiling. And just give you a little bit of background for our viewers. So the 16S gene is highly conserved. It's around 1,500 base pairs. And it consists of 9 variable regions. And it's these variable regions that we exploit to identify the various members in the community. That's as a sort of paternity test for the microbes in the community. And it allows us to classify down the genus level. And so this is amplicon sequencing. So what I've depicted at the top is PCR primers hybridizing to the V4 region. And due to the nature of next generation sequencing and the capacity, we can pool multiple samples into one sequencing run. And the way we do this is that we attach molecular barcodes to our PCR primers. So that allows us to bioinformatically tease out which members of that pool contribute what signal, so identifying which microbes are in that sample. So we typically pool around 400 samples in one given lyseq run. So what would 16S data give us? First, it tells us who's in the community. It gives us a relative abundance of those members of the community. And then it allows us to take a look at how those samples relate to like samples in this principal component plot that you see here. We have in this particular sample a smooth oral airways and generally. You see that they cluster nicely with their members. So let's shift gears a little bit and talk about the accreditation process. And so two key components of the accreditation process would be first to have a reference material that you could send out to laboratories across the globe to standardize or to evaluate the different laboratories and how they're processing the sample. And then you need to set up best practices so that we can even the playing field for those laboratories. And so we're going to specifically talk about those reference materials moving forward. So I'd like to talk a little bit about the CAP accreditation process. There's a number of organizations that you can employ to get accredited to use CAP. And so if you meet the minimum requirements, which would be to have a clinical lab director, sufficient facilities, qualified personnel, and validated instruments, and then you submit your application and your fee. They'll send you a checklist, which is 300 to 500 pages, which you must address line by line. So it's quite an endeavor. What I've done here is just kind of boil that down to the key components. And the first would be efficiency testing, which is really validating your laboratory as a whole. And then you have validation of instruments and equipment, competency testing, which is validation of your personnel, and then of course validation of the tests that you're offering. And so we'll talk about validation of our pipeline for 16 years. So the measurements that you would take in a CAP accreditation would be first accuracy, which is simply the agreement between the observed and you're expected. So you put a characterized reference material into your pipeline. Do I get the answer back that I would expect? The second measurement you would want to take precision and not reproducibility. So we look at that several different ways. One would be microbiome, so if we repeated samples within a given pool for 16S sequencing run, do we get the expected values back? And then the next would be an infer comparison. So if we completed sequencing on a pool and then we repeated that pool, do we get the expected results? So this also works for reference materials, specifically not communities, which is what we'll talk about in a moment. And then we would want to measure analytical sensitivity, and this is just the lower limit of detection. And so as we're talking about the mock community, we would spike in single isolates at lower concentrations at or near our limit of detection and see if we can evaluate that we're going to make it better. And then last but not least is analytical specificity. Can we correctly identify those members in the community? So one of the things that we would do is choose light strains or common strains and see if we can identify those in the context of a matrix. So this slide tells us, it's a snapshot of the validations that we ran in our accreditation process. And so at the top would be the facility as a whole. And so we would sequence a pool at the CMMR and then we send that to the same pool to the Community Sequencing Center here in Baylor and have them sequence, and then we compare the two data sets. Next would be validating the sequencing pipeline. So we would simply, as I indicated earlier, simply repeat a sequencing of the pool. So that would be CMMR versus CMMR. And then we want to evaluate our technicians. Is there any variance between technician 1 and technician 2, which would be our intra run variance. And then we have a fully automated pipeline from extraction through sequencing setup or library prep. So we need to evaluate any variance introduced through that automation. So we would complete a manual setup of a pool, sequence that, and then compare that to the automated setup. So we know through our validation studies that extraction introduces the greatest amount of variance, sample extraction. We have a number of different extraction kits that we utilize in the pipeline. One would be the mobile power flow, and another the mobile power mag. So we compared those two in a similar manner where we repeated pools and our reference materials. Another validation that we want to complete would be a low quality run. So at the end of a MySeq sequencing run, if we have any quality matrix, one is a P30 score, and then alignment and output. So if we had a sequencing run that was low in any of those metrics, we would then repeat that sequencing run and then compare those two data sets. And then we need to validate our analysis, and that would simply be taking a raw data file, which is an ASCII, and then repeating the analysis, recording the versions of the software pieces that are used in that analysis pipeline. And then last, proficiency testing would be proficiency of the laboratory. So we would submit blinded samples or reference material to our pipeline and then evaluate that as compared to the previously run of that reference material. So now we'll switch gears a little bit and talk about reference materials. And so I'd like to define that, because I think there are parts of the definition that you keep in mind as we evaluate reference materials. So that is that a reference material in this context is any stable, abundant, and well-characterized specimen that is used to assess the quantitative or qualitative validity of the measurement process. So we break the reference materials, the type of reference materials we use in metagenomic sequencing down into 3 categories. I'm going to describe each. The first category would be environmental samples. So that is a sample from a given environment that you use as a reference material. So if you have a sample source that you're testing, you may take an aliquot of that and use that as your reference. So the key there is that we obtain something in high enough abundance to carry us through the study, keeping in mind that some of these studies may last over a year for prospective studies. We need to make sure that there's limited variability among the aliquots, and so you may homogenize the sample and then aliquot that in sufficient quantities to complete the study. And then lastly, that it be stable over long periods of time, so we need to evaluate the shortage conditions. So the benefit of an environmental sample would be composition, because it's taken from the sample source that you're testing. Obviously, it closely matches the complexity of that sample. Some of the drawbacks, though, are that because it is complex, it's difficult to characterize. So break that down is, do I know exactly what is in that community that I'm using as my reference material? And then a second drawback would be the lack of reproducibility. And so does that sample source change over time? For instance, if we were taking a stool sample from an individual as our reference material, as their diet changes, that sample source is changing as well. Likewise, if we were taking a soil sample, there may be degradation over time, the pH changes, which will change the makeup of that sample. So the second category would be an in vitro model, and this would be the use of a bioreactor to cultivate microbes. We'll talk in the subsequent slide, we'll talk a little bit more about how we can utilize that for different types of reference materials. And so the benefit of this is that you can control the environmental conditions of the bioreactor. You can control the community makeup. And then you have virtually unlimited supply to cultivate in the bioreactor. And then it's relatively reproducible. So the drawbacks is, the drawback for this particular model is that you're unable to truly reproduce the ecosystem. So we're moving away from the license to the food sample. And then there's the potential for batch-to-batch variance, so we want to control for that as well. So then the third category would be pure microbial isolates. So as the name indicates, these are pure strains of microbes that you buy from vendors in MBQC levels. The benefit is that pure isolates, so you know exactly what is going into your reference material, and that it should be well characterized. And then the third benefit would be virtually unlimited supply of individual capability. And then the drawback is that it is inherently artificial. So it lacks the complexity of, say, an environmental sample, but you gain characterization because you know exactly what you need to do. So this figure here given to me by Dr. Microbial, I think really summarizes those last 3 slides. And so if you look on the far left side, would be the environmental sample. So you would take, for instance, a stool sample, homogenize that aliquot, and then send that right to and so again, the benefit there is you get the complexity of the sample that you're testing. Moving over to the right, just one slide over, with the use of the bioreactor to cultivate a fraction of that environmental sample. And so you're moving away from the complexity that you had with the environmental sample, but maintaining some of the matrices that are in that sample. And then moving further to the right would be the use of the bioreactor to cultivate your isolates. And so we purchase strains, let's say top 20 strains that you would find in the environmental sample, place those in the bioreactor and set the conditions. And then further to the right would be the use of pure isolates. So we would cultivate those individually and then mix them in some ratio. So if you look at the bottom of the slide, you see that there's this trade-off between likeness to the true sample source and then reproducibility of moving away from towards the pure strains. I would add to this characterization. So it's very difficult to characterize on the far left side, whereas with the pure strains on the far right side, we're able to characterize what is going into our reference material so that we have that known quantity and we can compare that to our observation. So, what sources of reference materials are available to us commercially or in the laboratory? So at the very top of the slide would be previously analyzed samples. And so, as I was talking about the accreditation process, I would refer to previously analyzed samples. So we would take a pool of previously sequenced and we would compare that data to the previous one. So that's obviously every lab has access to that. Moving down the slide would be those references that were used in the human microbiome project. We had EBI and APCC strains, and then a reference material produced by Dr. Defoe and the RoboVet. Unfortunately, those reference materials are no longer available. There are a few commercial sources coming up there. So Sino Research has the Xynobiotics, which is available, and I'll show you some data from that reference material later. APCC has 2 reference materials that are not available now, soon will be, and these were developed with the ABRF, Metagenomics Research Group. So in the absence of a commercially available reference material, you might go out to develop your own reference material. And so some of the considerations that you would want to look at are taxonomic classifications. So you would want a broad spectrum that represents your test material. You would want to look at the growth conditions of those individual members, are they available? And then you would want to know reference sequence. For instance, is there a known copy number on target sequences? 16 S, 18 S. You would want to know the genome size, and then GC content, so you can account for any GC elements that you have. And then if there's functional information, you can inferences to what's going on in the community. So switching gears a little bit, I want to talk about the mock community that we created. I was asked to speak at the Standard for Microbiome Measurement workshop on this past August. So I was asked to talk about the accreditation process, but I also wanted to talk about reference materials. And so we identified a project for that particular workshop. So what we did was we used the LabSite's Echo 525 to assemble our mock community. And the Echo, for those of you who don't know, is an acoustic liquid transfer robot, and so it transfers nanoliter quantities of liquid from source plate to a destination plate. So it does this with great precision. So we thought that it would be a great tool to assemble these mock communities in some very specific ratios with great accuracy. And so to validate this, we would want to look at a manual setup as compared to the use of the automated setup with the Echo. So we produced both a whole cell mock community, and and then a companion genomic DNA mock community. And the reason for this is we wanted to look for any variances in extraction efficiency in those members of the mock community. And so some of the variables that we looked at were size, shape of the microbes, and then also the growth media, and how that would impact our ability to transfer using the method. So the measurement that we would want to take is we would need a cell count both prior to transfer and post-transfer. And then likewise, we would want to quantify the DNA companion mock community, both prior to and post-transfer. And some of the variables that we looked at there would be genome size and DNA concentration, which could impact the transfer. So on paper, this is like a slam dunk project, and we were in a limited time frame leading up to that workshop. So some of the challenges that we encountered were our ability to quantify the microbes prior to transfer. So we looked at plating and CFU counting, which is laborious and cumbersome, so that didn't work out well for us. Use of a hemocitometer, again, laborious. And then the use of spectrophotometry. The concentrations that we needed for our mock communities, it just wasn't accurate, didn't give us the accuracy that we needed. So that was that one. And then the folks at the lab site suggested that we use luminescence as our measurement. So we utilized the Omega backpatter flow, which worked with some success. But we were really lacking a very accurate means of quantifying how many microbes we're putting into the mock community. And then secondly was our ability to accurately quantify sub-nanogram quantities of genomic media. So down at that concentration, spectrophotometry, specifically with the use of the nanodrop, was out. Fluorescent measurements were insufficient, so we tried to look at qubit. So then the obvious choice would be PCR. So we have the capacity to do that in the laboratory. Unfortunately, as we chose the members of our mock community, our team set out to identify all of the metadata associated with those members, and one would be copy number of the 16 estimates as we were using as our measurement. And we had the PCR assay designed for 16 mass. And what we found is there's not a properly annotated database with 16 mass, and so we've got numbers back that vary 6 to 10 or 10 to 20 copies. And so in order to do the math and identify how many genomes we're placing in the mock community, we need an exact number of 16 as to whatever our target genome would be. So in light of those issues, we pressed forward, the transfer would be echoed and evaluated our mock community. So the first tool that we used was Curtis Heppenhauer and Harvard's metaplan. And lo and behold, if you look at the lower right, which is the output from metaplan, we got back the correct IDs of a number of our mock community and also an appropriate ratio. So the next thing that we want to do is evaluate the mock community just as we would a normal 16S sample for our sequencing outcome. So we utilize the Tina, which is a software tool that was developed by the bioinformaticians in our group. So we have a group of 5 bioinformaticians who do our analysis. So this tool is available at the website listed at the bottom. And you'll see the output. So here's the results from our mock communities. On the left side is the commercially available Zymo Research mock community, which is Zymo. We looked at relative abundance, and so we had 3 technical replicates, and you can see in this bar chart that they're highly reproducible. And then on the far side would be our mock community that we assembled with the echo. Again, you see it very reproducible in these technical replicates. So the next thing that we could look at is data diversity. And so this is a principal component analysis of that data set. And you'll see the light-colored balls plotting very closely in 3D space to one another. So again, very, very reproducible. So it makes a very pretty picture, but auditors want to see numbers to support that. So we plotted the distance matrix, and so on the left-hand side, on the y-axis, you can see the automated sample. The x-axis, obviously, the main setup, and the distance matrix shows that how reproducible these assemblies were. So some of the future challenges for developing reference material for metagenomic sequencing would be we need some advances in technology. So the difficulties that we encountered in this project were our ability to accurately count microorganisms. So the techniques that we used weren't sufficient, they lacked the accuracy that we need. Secondly would be the ability to accurately quantify subnanogram quantities of DNA. And then thirdly, we need an accurately annotated database to review the reference genomes that we utilize standards. And then thirdly, an accepted set of measurement standards so that we can distribute the various laboratories across the globe. And I don't think there's a one-size-fits-all. Community is great. We also need to look at environmental control, possibly having in vitro models into this as well, and then usage for microbial. And then last but not least, there needs to be some best practices. We know that sample collection and handling is the greatest amount of variability, so best practices on that help standardize the field study. Extraction method is, again, it's a lot of variability, so practices on that. And then finally, analysis parameters, so everyone utilizes their own software tools in their pipeline. So some ability to standardize those settings. So with that, I'd like to acknowledge my partners in this, Dr. Alan at Gulf University for finding the information on the bioreactor, and also for helping some of the mock communities. Sam Corey at MIS, Russ and Austin Walker at LabSite, now is in San Diego, with Rob Knight for their contribution to that project for the MIS workshop.
Thank you, Dr. Carmichael, for that informative presentation. It's time for Q&A. If you have a question you'd like to ask Dr. Carmichael, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the Send button. We'll answer as many questions as we have time for. If we are unable to get to your question, Dr. Carmichael will follow up with you via email after the presentation. Let's get started. Our first question is, what is the greatest source of variability in the metagenomics pipeline?
So in our accreditation process, we validated every step of that process, and so what we found is the greatest variability is in the base bar. samples. So sample handling, and then also when a sample is collected, so from the time of collection to the point of freezing the sample or adding something to the surrogate. And also compounded by the different extraction kits. And so we tried to standardize that for our lab and for our commercial partners that are collecting through, say, a CRO. We give them very strict guidelines on how to collect those samples.
Great, thank you so much for that answer. The next question is, what sets the Echo apart from other liquid handling devices for reference material assembly?
So the great thing about the Echo is not only the ability to transfer nanoliter quantities of liquid from source plate to the destination plate, it's the accuracy and precision that it does that. So because the volume associated with it is that manual hand pipette, you just can't get to that level of accuracy. And then secondly is that what we would call dynamic assembly. And so if you had a say a 384 well plate with its microbes, you could cherry pick those microbes and assemble the mock community or the reference material to suit your specific needs. And so you would want to create a mock community that is as similar to your test material as possible. So you can envision a vendor such as APCC having numerous 384 well plates full of microbes, and then custom design a mock community for your specific research interest.
Great, thank you. What type of reference material do you think will be most useful as metagenomics matures?
Well, again, I don't think that there's a one-size-fits-all. I think there will be a modular reference material. So, for instance, if you were looking at the skin microbiome, you may select the top 20 microbes that's commonly found in those sample sources, and then you may add to that, or it may be it's that you utilize a mock community and an environmental sample. So 100 different skin samples, and then homogenize those and output those as reference material, your best source of reference material. You capture both the complexity of the sample that you're testing, but then a well-characterized component.
Thank you. And we'll wrap up with one more question, and that is, what is being done to assure smooth transition from bench science to clinical diagnostics for metagenomic studies?
So this is up to the research community to fulfill. And so there's a number of organizations, you mentioned in my intro, IMSA, and so we are identifying reference materials that are available now, and then also efforts towards building reference materials. Also through the accreditation process, we had to develop our own proficiency tests so it's not commercially available. And so they've agreed to look at a reference material that we would submit as a commercial commercially available reference material. Groups like ABRF and their research groups within that organization are developing mock communities, and so you see two of those that are out that will be available through AECC.
Thank you. I would like to once again thank Dr. Carmical for his presentation. Do you have any final comments?
Thank you, Julie. And questions that we didn't get to, I'll answer those online.
Thank you once again, Dr. Carmical. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through July of 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.